So line 23A13, Halophilic dentrifying bacteria prokaryotic alien DNA Salt Lakes Wow Seti. There's the alien radio signal there. Okay, again, a lot of keywords for this particular video. Extremely halophilic dentrifying bacteria, hypersaline inland lakes alien DNA, molecules, hypersaline habitats, inland salt lakes, sea salterns, environment, heterotrophic, haloarchaea, NaCI concentrations, 3M, Oren, 2002, that's Oren, hypersaline prokaryotic communities, halophilic acteria, functions, anton, sorokin, chemolithotrophic bacteria. Why do they have to give them such difficult names? Why can't they make it easy for the rest of us? So SOB, high energy. No, I'm not saying it's an SOB. That's the acronym. I don't know what it means, but just so you know. High energy yield sulfide, thiosulfate, Oren, O-R-E-N. It makes me think of Oreo cookies. 1999. Genes, sulfur-oxidation pathways, molecular Friedrich culture strains. Keyword research, line 23, Wow Seti data. Finally. So July 2012, almost a year and a half ago, because we're in 2014 now, in April. Google extremely halophilic dentrifying bacteria, quotes and PDF file results from a blog. So extremely halophilic dentrifying bacteria from Hypersaline inland lakes, and that's from ijs.sgmjournals.org. It's a PDF file from D.Y. Sorokin in 2006. Extremely halophilic dentrifying bacteria from hypersaline inland lakes, halovibro, denitrifications SP, NOV, and halospina defecation G, and whatever. And it's at googles.ca. Hypersaline habitats such as inland salt lakes and salterns were traditionally regarded as a low diversity environment dominated, and it doesn't say, yeah, okay. Were traditionally regarded as a low diversity environment dominated by heterotrophic haloarchaea, which grow optimally at NaCI concentrations above 3M, or in 2002. Now it is becoming clear that hypersaline prokaryotic communities may also include extremely halophilic bacteria, which might have important functions in these unique habitats. Anton LL 2002 and Sorokin 8L 2006. Among the chemolithotrophic bacteria, SOB, there's the SOB there, have the best chance to adapt to extreme conditions. So those SOBs have, sorry, I had to make a joke. Those SOBs have the best chance to adapt to extreme conditions. Don't we all wish we could? Such as high salt, owing to a very high energy yield available during complete oxidation of sulfide thiosulfate to sulfate. Oren, 1999. Now all of you are going to want to go buy a bag of Oreo cookies. Oren, you know, get it? With your spot of tea. However, so far, no culturable SOB phenotypes with the same salt dependence. Yeah, you know how people get the giggles when they film me, can't stop. As haloarchaea have been described, since functional genes of sulfur oxidation pathways are not conserved and have only recently started to become a subject for molecular analysis, Friedrich al. 2001-2005, the culture-independent approach is not yet available for diversity analysis of SOB. Therefore, traditional methods of enrichment and isolation in pure culture remain the main approach. Once strains are isolated, group-specific phylogenetic probes can be developed to detect the cultured SOB in their natural habitats. According to the origin, hypersaline aquatic habitats are divided into two major types. The best studied type is the sea solar salterns, thalassic, which are relatively easy to access and have an advantage for investigators in offering a whole range of salinity gradients within a short distance, various stages of evaporation. Most of the data on microbial diversity of halophiles have been obtained from these habitats. Less studied are the athelistic inland hypersaline lakes, located mostly in areas with evaporative climate, primarily evaporates or sometimes formed by a gradual dissolution of ancient salt deposits, secondary evaporates. The major difference between the two major types is the much higher magnesium content in the thallasic brines and usually the higher sulfate content in inland lakes. In our search for halophilic SOB, we mainly focused on inland hypersaline lakes, but also used samples from a sea saltern and from a deep sea salt brine formed during dissolution of ancient salt deposits for comparison. The analysis demonstrated unexpectedly high diversity of hamophile SOB inhabiting various types of hypersaline environment. And that's from mic.sgmjournals.org. And the data will continue in the next video. And thank you for watching and giggling along with me.